This is not a prokaryote, this is a eukaryote. Why? Because your messenger RNA here, at the end of it you have a poly A tail, and then at the beginning of it you have a 5' cap. Okay, so here is just giving you the picture of what the initiation of the eukaryote will look like. And also it's important that the proteins from the cap, from the 5' cap, and also the proteins associated with the poly A tail are important for the assembly of the large and small subunits in the ribosomes in the eukaryotes. For the translation process, this is in the eukaryotic system. We see how the prokaryote will start the translation, almost, ready to start translating. Now, if you compare the prokaryote to the eukaryote, let's see what happens to the eukaryotes here. So for the initiation, you have to get, again, the ribosomal subunits ready. So you have here the messenger RNA that will bind to the small ribosomal subunit, just like it did in the prokaryotes. So now in the eukaryotes, you have this complex formation also of initiation factors. You don't have a particular initiation factor that is more important than another like you had in the prokaryote. Here you have a collective amount of initiation factors that's called IF4. So that will bind to the 5' cap in the eukaryotes at the 5' end of the messenger RNA, and it will interact also with IF3, which is in the small ribosomal subunit, and interaction of initiation factors are going to help to assemble the small and large subunits together here. Now, for the eukaryotes, you don't have that shiny organelle sequence. You have this other sequence, this other code here that is called the Kozak sequence. That it comes also just before the start of translation signal, before the AUG. So you have a lot of things that are similar and some things that are different. Of course, you always have more proteins that are involved in the eukaryotic system, and you always have more protein interaction that is needed for initiation here. So once you have here the assembly of the small and the large subunit at the 5' cap, what happens here is that the large and small subunit, or these ribosomal subunit here, are going to start scanning the messenger RNA to see where it's going to start the translation into protein. So once it scans, it goes through the RNA here, it finds the AUG. So at that time, it's going to start the translation. So here you have some scanning process. And then in the prokaryotes, it was a simpler type of process. You have the shiny organelle sequence, start is right there, right after the sequence. So you have the initiation here, you have the large subunits, they'll bind to the complex, and you have also GTP that is used for energy, and that once you hydrolyze the GTP, you, just like in the prokaryotes, you kick out the initiation factors, and you have this system here that is ready to synthesize protein. Now for the elongation. Again, you have the sites that you need to remember for the ribosomes. You have the exit site, the peptidyl site, or processing site. Now, if you don't remember, if I ask a question and say, what is the sequence of protein synthesis in the ribosomes, and say, oh, no, there's a P site, I don't remember what that is for. You keep in mind what is happening, keep an image in your head of what the molecules are doing. So P is something processing, is going to bind some type of amino acid there, is going to form some type of polypeptide chain. You know that you have A site that's going to accept tRNA, P site is going to process it, E site is going to kick it out. You also have elongation factors that are important to keep the system going, and those are TU, TS, and G factors that you're going to see here too. Okay, so for the elongation, the tRNA gets into the A site. Now the methionine will to the P site, so it will allow for the next tRNA to bind to the A site. Okay, we're going to see this into more details, how the peptide bond is formed. You have here at the P site, the chain, in this case, it is only one, right? This is the methionine right there, AUG. So it's only one amino acid here. It's on the P site. Now on the A site, you're going to have your tRNA that is coming next. Now here, you have two amino acids that are lined up.
See, it's for glue. That's okay. See?
So for this elongation, you need these elongation factors. So here you have the elongation factors TU. So next, elongation factor 2 is going to help out for this peptide bond here that's going to synthesize. What happens at the P site? This is going to be joined to the A site amino acid. So it's going to be from the P to the A that you're going to transfer this amino acid. Now it makes sense because right after the P site is your little exit site. So in order for this tRNA to get out of there, it cannot get out of there carrying the polypeptide chain. It has to get out of there being empty. The peptide chain has to stay in the system because it has to be elongated. If the P site is carrying the peptide and then it gets kicked out, what are you left with? Nothing. So the chain has to stay in there. In order for the chain to stay in there, this has to go from P to A. And then the empty tRNA gets in there. See, empty, no amino acid. And then it gets kicked out. Polypeptide will grow here at the A site. Now the tRNA that is at the A site has to be shifted to the P site so that the A site can accept the next tRNA.
So wait, are they doing it in pieces? Like, I don't really understand. What are they kicking out?
Okay. Let's go through some more details. We're also going to have more figures. But you imagine, large subunit is on top of the small subunit. Messenger RNA is in the middle. The 5' end of the messenger RNA is right here, which means start is going to be right there. So AUG start. What does that mean? tRNA that has anticodon to AUG gets in there and say, okay, I'm going to start protein synthesis here. P site. Now on the A site that is going to be empty here is going to get another tRNA that's going to be anticodon to this codon right there.
Okay.
And now this other tRNA is going to carry another amino acid.
Okay.
Now you have both sites occupied. Both of them have a tRNA and also an amino acid.
Right.
Now, how is it that you're going to make your chain? You have to make a chain of protein, right? You have to go from mRNA to protein. Here, you're going to have to connect one amino acid to the next amino acid. The P site tRNA kicks out this amino acid to the A site, right there. Okay, you're going to see here, and then the two amino acids are connected there by a peptide bond. Now, once this tRNA gives its amino acid to the A site, now it's empty. It does not have the amino acid there anymore. So now it's ready to leave the system. So it gets kicked into the exit site and gets kicked out of the system. Now this new tRNA here that has two amino acids together, it needs the next one. It needs to leave the next codon. So this whole system of large and small subunit are going to shift 3 bases to the next one. Okay, it doesn't shift one base, so the whole system goes 3 bases to the next codon.
Okay.
It knows how to do the square dance.
Okay.
3 bases. Now here, when it's on the P site, it's going to become this with another peptide here. So the next codon here is going to get its next tRNA that's going to carry another amino acid. Okay, on the A site, we just went through there. See here? We have elongation factors that are helping with the peptide bond formation. Again, this process is forming a covalent bond. It uses energy. So elongation factor plus GTP. The energy that is used here is not ATP, it's GTP. Okay? It uses energy to make the peptide bond. Now, remember, this tRNA that kicked out was empty. So this whole chain shifted to the P site. Now A site is empty. Now codon here is going to call for another tRNA to come in there. Okay, just remember, A site is empty, it's going to call. Okay, bring me tRNA that has my anticodon. tRNA comes in there, and again, you're going to start the same process again. The P site is going to shift all of its tail of polypeptides to the A site for another peptide bond. And then the P site is going to be empty. tRNA is going to shift to the exit and get kicked out. And then the ribosomal subunit, big and small, will do the dance, shift 3 to the next one. and call for the next tRNA. Okay, going to the A site. And the thing happens over and over and over again until you get your whole peptide done. Now, when you get at the end of your polypeptide chain, you have to finish it somewhere. You will have one of these codons. You are going to hit one of those codons that do not code for anything. So it's not going to code for a tRNA. It does not code for any amino acid. So what's that stop codon going to do? It's going to code for termination factors. They're going to get into this A site, and they're just going to disassemble the whole thing. So that's a termination factor. It's just a more detail on the elongation. You have elongation factor 2 that comes with some energy and helps to elongate that amino acid chain here. It's going to help to make this peptide bond and move the whole system one codon down the line. Okay, see here, you have the elongation factor 2 here. After you hydrolyze GTP, you get GDP and phosphate. So with that hydrolysis, you have the energy to make that peptide bond. So now after the use of this energy with the elongation factors, you get this chain that is starting to grow out of that ribosomal unit. Okay, those are just some repeats. The one that is empty gets kicked out, and it goes over and over. I just put it again many times, so when you're studying it, you see so many times that you totally memorize it.